Welcome to Deep Dive Lab, where we break down and dissect cutting-edge scientific research. From advanced biomedical technologies to breakthroughs in AI, material science, chemistry, and physics, all told as compelling, easy-to-follow stories.
Glad to be here for this one. It's a really wild topic today.
It really is. Today, we are looking at something that feels like it belongs in a sci-fi movie, but is actually fundamentally reshaping pharmaceutical labs right now as we speak.
Oh yeah, it's happening right now.
So think about Han Solo being frozen in carbonite at the end of The Empire Strikes Back.
Right, classic scene.
Right. And the whole point of that carbonite is to preserve him perfectly. But the freezing process itself is incredibly violent and risky. I mean, he comes out of it blind and disoriented, suffering from, hibernation sickness.
Which makes sense because freezing is actually really destructive to biology.
Exactly. But now, imagine if being frozen in that carbonite didn't just freeze him, but actually systematically rearranged his internal organs while he slept, giving him an entirely new anatomy.
It's a completely wild thought experiment. I mean, the baseline expectation in biology, and really in all of cold storage preservation, is that the preservative environment is completely passive.
Like, it's just there to hold things still.
Exactly. You put something in a deep freeze to just, hit the pause button on molecular degradation. You definitely don't expect it to completely rebuild the anatomical structure of the thing you're trying to save.
And that brings us to the strange, real-world scientific mystery we are dissecting today. We're looking at advanced microscopic medicines, specifically mRNA-Lipid nanoparticles.
The heavy hitters of modern medicine.
Right. So the core question driving our deep dive today for you listening is this. Why does simply changing the chemical water that a microscopic medicine floats in completely alter whether it cures a disease or morphs into a useless, leaky pile of mush the moment it gets frozen?
It's such a bizarre phenomenon.
It really is. And to answer this, we're pulling from a fascinating breakthrough paper published in the journal ACS Nano.
Yeah, this is a phenomenal piece of research.
The research was spearheaded by Mohammadi-Zerankeshi, Daniel Estabrook, Alexander Maris, and a highly collaborative team across the University of Texas at Austin, Eli Lilly and Company, and Boston University.
A real powerhouse team.
For sure. Our mission today is to understand how these researchers prove that the liquid a medicine is stored in isn't just passive packaging. It literally acts as an active architect, rebuilding the medicine from the inside out.
And, to appreciate the magnitude of what this team discovered, we really first have to understand the microscopic medicines themselves. We are talking about mRNA-lipid nanoparticles, or mRNA LNPs.
Right, which most people know from the last few years.
Exactly. For anyone who has followed the development of the COVID-19 or RSV vaccines, this is the underlying technology. At a high level, these are incredibly sophisticated, microscopic fat bubbles engineered to deliver delicate messenger RNA directly into our cells.
But it's not a perfect system, right?
Not at all. This delivery system is plagued by two massive systemic weaknesses that formulation scientists have been battling for years.
So I would assume the first major bottleneck is getting the medicine to actually function once it is inside the body. Like, we know that when a cell encounters one of these lipid nanoparticles, it doesn't just passively let it float inside.
No, the cell actively swallows it up. It wraps its membrane around the nanoparticle, pulling it into a specialized cellular compartment called an endosome.
And the endosome is basically the cell's internal recycling center, right? Or like its garbage disposal.
Yeah, that biological sorting mechanism is brutal. Once the nanoparticle is trapped inside the endosome, the cell activates these specialized protein pumps, specifically VAT pases that rapidly pump protons into that compartment.
Just flooding it with acid.
Exactly. This drops the pH significantly, creating a highly acidic environment designed to just digest whatever is inside. So for the mRNA to actually do its job and trigger a therapeutic response, it has to physically break out of that endosomal garbage disposal and escape into the main fluid of the cell, the cytoplasm.
And how often does it actually make it out?
Well, currently, the failure rate is staggering. Upward of 90% of the mRNA gets permanently trapped in the endosome.
Wait, 90%?
Yeah, it just gets degraded by the acid and enzymes and never actually works.
That is a staggering loss of efficiency. You are basically flooding the body with a massive excess of medication, just hoping a tiny fractional percentage manages to escape the cellular garbage disposal.
Exactly. That's the biological hurdle. But these advanced therapies also have to be manufactured, stored, and shipped globally.
Oh, right. I remember during the vaccine rollouts, the logistical nightmare was keeping these vials in ultra-cold freezers at -80 degrees Celsius. So the physical freezing process itself has to be the second major vulnerability.
It is entirely destructive. Freezing and thawing these nanoparticles is a violent mechanical and chemical ordeal.
Because of the ice crystals.
Yeah, in standard storage liquids, the freeze-thaw cycle routinely causes the nanoparticles to rupture. They lose their structural integrity, they aggregate, meaning they just fuse together into massive clumps, and they leak out all their delicate mRNA payload.
So they just get shredded. But what did scientists think was happening? Like, what was the original assumption here?
Well, until recently, the scientific consensus was that the storage liquid, what chemists call the buffer, was just an inert background player. The standard assumption was that the buffer's only job was to maintain a stable pH so the chemical components didn't degrade at room temperature.
Okay, let's unpack this for the listener. It's like making a perfect smooth soup. Right. You put it in the freezer, and when you thaw it out, the fat has separated into gross, hard clumps, and the flavor is totally ruined.
That's a great analogy.
Mhm.
So formulation scientists were just mixing up their high-tech mRNA and specialized lipids in whatever standard industry-approved buffer they had on hand.
And the data from the labs must have been incredibly confusing.
Oh, it was a nightmare. You can mix a fresh batch of nanoparticles, test them immediately, and see stellar results. But take that exact same vial, put it in the freezer overnight, thaw it out, and suddenly the therapeutic efficacy drops to absolute zero.
That is wild.
The frustration in these labs was palpable. The physical stress of the ice crystals and the changing thermodynamics of the freezing process were clearly destroying the architecture of the nanoparticles. But no one understood the underlying mechanism of why certain batches survived slightly better than others.
Or why the failure rate was just so unpredictable.
Right. And that is the exact wall of confusion the team at UT Austin, Eli Lilly, and Boston University decided to tackle. They stopped treating the buffer as a passive swimming pool and started interrogating it as an active, independent variable.
So how did scientists figure this out? Because this is where the story gets really interesting. They set up a brilliant comparative investigation, right?
They really did. They took identical mRNA LNPs, meaning the exact same mRNA payload, the exact same structural lipids. They even tested 3 different industry-standard ionizable lipids, known as LPO1, MC3, and SM102, just to ensure their findings were universal.
Right, to make sure it wasn't just a fluke with one type of fat bubble.
Exactly. And they took these identical nanobots and suspended them in 3 different widely used chemical buffers: tris, histidine, and citrate.
Okay, so tris, histidine, and citrate. The exact same medicine, just floating in 3 different types of water.
Yep. And when they tested these formulations in their fresh state, meaning refrigerated but never frozen, a massive discrepancy emerged immediately. The nanoparticles stored in the citrate buffer vastly outperformed the others.
By how much?
When introduced to human cells in vitro, the citrate-formulated LNPs demonstrated up to 151% higher transfection efficiency.
151% increase. That is massive. That means over double the amount of mRNA successfully breached the walls of the cellular garbage disposal, escaped into the cell, and actually did its job.
Yes, exactly.
But the nanoparticles themselves were identical. So to understand how simply swapping out the surrounding liquid caused such a drastic spike in performance, they had to look at the internal atomic structure of the nanoparticle itself.
And you cannot just put something this small under a standard microscope.
Right, it's way too tiny.
The researchers utilized a highly advanced technique called small-angle X-ray scattering, or SESS. It basically involves firing a focused beam of X-rays at the sample.
So they're using X-ray vision on it.
Basically, yeah. As the X-rays hit the electron clouds of the lipid molecules inside the nanoparticle, they scatter. And by mathematically analyzing the angles and intensities of that scattered radiation, scientists can reconstruct the precise geometric arrangement of the molecules inside down to the nanometer.
That is so cool.
It is. They use this SAXS data to watch how the internal geometry of the nanoparticle changed as it was exposed to the acidic environment of the endosome.
Okay, so let me see if I have this right. When the endosome gets acidic, the unique ionizable lipids inside the nanoparticle react to that acid. They absorb the incoming protons and take on a positive electrical charge.
Yeah.
And that sudden shift in charge changes the physical shape of the individual lipid molecules, right? It widens their tails so they shift from a cylindrical shape to a conical shape.
You've got it. That geometric shift at the molecular level forces the entire internal architecture of the nanoparticle to reorganize. The internal lipids transition from a disordered, tightly packed sphere into a highly structured, complex geometric pattern called the inverse hexagonal phase. or the HII phase.
The HII phase. So instead of thinking about this structure like a microscopic drill bit mechanically punching a hole in the cell, we should visualize this process more like two soap bubbles.
Oh, I like that.
Yeah, so when you press two soap bubbles together, they don't immediately pop each other. Under the right conditions, their surfaces merge, creating a shared wall or fusing into one larger bubble.
Yes.
So the inverse hexagonal phase essentially lowers the energy barrier for that merger. The conical lipids flip outward and link up with the lipid membrane of the endosome itself.
That is the perfect mental model. The nanoparticle membrane and the endosome membrane physically fuse together, creating an open pore, a doorway that allows the mRNA payload to safely spill out.
Okay, so how does the citrate buffer fit into this?
Well, what the X-ray scattering data revealed was that the citrate buffer actively forces the excess lipids inside the nanoparticle to transition into this crucial inverse hexagonal phase much earlier in the acidification process.
Oh, so it speeds it up.
Exactly.
Huh.
Citrate molecules have specific chemical properties that help shield the repulsive forces between the lipids, allowing them to snap into that doorway building structure faster and with less energy. Because it morphs earlier, it escapes before the endosomal acids can permanently destroy the mRNA.
Okay, wait, I have to push back here. If citrate makes these nanoparticles 151% more efficient at building that escape hatch, logic dictates we should just use citrate as the universal standard for all mRNA therapies. Like the biological bottleneck is solved. Let's call it a day.
I mean, you would think so, right? But this brings us crashing right back into the second major vulnerability, the freezing process.
Oh, right, the carbonate freezing.
The freezer is where the entire citrate paradigm completely collapses. When the researchers took those high-performing citrate nanoparticles, placed them in a standard minus 80 degrees Celsius ultra-cold freezer, and subsequently thawed them, the structural analysis showed complete devastation.
What happened to them?
The nanoparticles had ballooned in size by over 918%.
Over 918%.
Yeah. They transformed from tightly packed, nanometer-scale delivery vehicles into massive, fused, disorganized clumps. Their structural integrity was shattered, the mRNA cargo was lost, and their ability to transfect cells dropped to virtually zero.
Complete failure. But wait, if citrate is an inert buffer at room temperature, the physical act of freezing the water must be altering the chemistry of the citrate itself, right? Because when water freezes, it forms a pure, rigid ice crystal lattice.
Exactly.
And that lattice is highly exclusionary. It physically pushes out any dissolved salts, impurities, or buffer molecules as the ice expands.
You are describing the physics of cryoconcentration, and it is the exact mechanism that destroys the medicine. As the pure ice forms, the dissolved citrate molecules are aggressively shoved into microscopic, unfrozen pockets of remaining liquid between the ice crystals.
So they just get squished together.
Yeah, the concentration of citrate in those tiny puddles skyrockets. This extreme localized concentration causes the pH in those pockets to plummet.
Wow.
Furthermore, citrate molecules carry a -3 electrical charge. As the pH drops and the ionizable lipids on the nanoparticles take on a positive charge, the highly concentrated, negatively charged citrate acts like a powerful chemical glue.
Oh, I see where this is going.
It bridges the positively charged surfaces of multiple distinct nanoparticles, permanently fusing them together into useless, aggregated masses.
So the very chemical property that made citrate so effective at membrane fusion in the cell makes it a catastrophic liability in the freezer.
It's a tragic irony, honestly.
But this failure is exactly what sets the stage for the true hero of this investigation, the Tris buffer.
Yes.
Because while Tris didn't generate the massive 151% spike in fresh performance like citrate did, it survived the brutal physics of the freeze-thaw cycle almost flawlessly.
Beautifully, in fact. The Tris nanoparticles maintained their precise microscopic size, they perfectly retained their encapsulated mRNA, and their therapeutic efficacy remained intact after thawing.
So how does Tris survive when citrate just turned everything to mush?
Tris acts as a formidable, multi-layered cryoprotectant. At a molecular level, the chemical structure of Tris includes hydroxymethyl groups. These groups are highly effective at forming robust hydrogen bonds with surrounding water molecules.
Okay.
And by aggressively bonding with the water, Tris molecules physically interrupt and slow down the formation of sharp, damaging ice crystals. They disrupt the ice nucleation process that would otherwise exert crushing mechanical stress on the delicate fat bubbles.
So Tris is essentially acting as a molecular antifreeze, softening the thermal dynamics of the freezing process.
Yes, and it also acts as a chemical scavenger. sweeping up harmful byproducts like aldehydes that naturally form over time and chemically degrade the delicate mRNA strands.
That is so elegant. But the Boston University and Eli Lilly teams didn't just document that tris was protective, they pushed the environmental variable to its extreme. They systematically cranked up the concentration of the tris buffer, didn't they?
They did, and this is where we get our real wow moment.
Hmm.
The standard industry-accepted concentration for tris buffer in a formulation is roughly 10 millimolar. The researchers escalated this to 50 millimolar and then pushed it all the way up to 150 millimolar.
It's just totally flooding the system.
Exactly. And the high-resolution imaging, specifically cryogenic electron microscopy, where the samples are flash frozen in a thin layer of vitreous ice and bombarded with electrons, it revealed a massive, unprecedented structural transformation.
What did they look like?
Well, at the standard low concentration, the nanoparticles look like typical uniform spheres, with the mRNA and the structural lipids densely tangled together in a solid core. But at 150 millimolar tris, the physical anatomy of the nanoparticle changed completely.
Like they grew arms or something?
Sort of. They grew these highly distinct, bulging, asymmetrical pockets on their exterior surface. In the literature, scientists refer to these protruding anatomical structures as blebs.
Blebs. I love that word. Okay, let's give the listener a visual analogy here. Imagine a standard lipid nanoparticle is a chocolate chip cookie. You've got the dough, which is the structural lipids, and the chocolate chips are the mRNA mixed evenly throughout.
Okay, I'm with you.
When exposed to the high-concentration tris buffer, it's as if an unseen force reaches into that cookie, extracts every single chocolate chip, and packs them tightly into one single gooey pocket bulging out of the side of the cookie.
That is brilliantly put. That distinct aqueous blister containing the isolated mRNA is the bleb.
Okay, so if the cryo-EM imaging gives us visual confirmation of the bleb, but doesn't explain the atomic physics of why the tris buffer forces the mRNA to migrate into that isolated pocket, the team must have turned to molecular dynamics simulations to track the physical forces, right?
Exactly. The researchers at Boston University built a massive atomic-level virtual model of the nanoparticle system. Molecular dynamics simulations don't just generate pictures, they use advanced force fields to calculate the precise Newtonian physics, electrostatic repulsions, and atomic attractions of millions of individual atoms over femtosecond time intervals. They literally watched the molecules interact in simulated real time.
Did the simulation show the tris molecules physically wedging themselves into the lipid structure?
It showed that tris is an incredibly persistent chemical wedge. The simulations revealed that the high concentration of tris molecules aggressively infiltrates the surface of the lipid nanoparticle. They form dense networks of hydrogen bonds with the functional head groups of the ionizable lipids.
So they just flood the lipids with bonds.
Yep. By saturating those lipids with hydrogen bonds, the tris effectively outcompetes the mRNA. It weakens the fundamental electrostatic attraction that was keeping the lipids and the mRNA tangled together in the core. The tris systematically pushes the mRNA aside, forcing it to pool together into that distinct water-filled bleb on the perimeter.
Wow. So the buffer is actively acting as a biological architect, completely reorganizing the internal furniture of the nanoparticle.
Exactly.
And the segregation of that mRNA into the bleb that solves multiple downstream problems simultaneously, doesn't it? First, from a preservation standpoint, isolating the mRNA in an aqueous pocket protects it.
Right, because when mRNA is tightly tangled in a dense lipid core, the lipids can inadvertently react with the RNA strands over time, forming chemical adducts, essentially binding to the genetic code and rendering it unreadable by the cell.
Which makes the medicine useless.
Right. So the bleb acts as a protective sanctuary, preventing those chemical collisions.
That segregation is crucial for shelf life. But the second consequence of the bleb is arguably the most revolutionary finding in the context of the endosomal escape bottleneck we discussed earlier.
A garbage disposal.
Exactly. Because the high concentration tris has forced the mRNA into its own isolated pocket, the rest of the nanoparticle is suddenly liberated. You now have a massive, dense domain of highly mobile excess lipids that are no longer anchored down by heavy mRNA molecules.
Which means when that specific nanoparticle gets swallowed by the cell and exposed to the acidic environment of the garbage disposal, those liberated lipids are free to rapidly shift their geometry.
Just like the soap bubbles.
Yes, they can snap into that inverse hexagonal phase, form that soup bubble-like fusion pore with the endosome wall, and guarantee that the highly protected bleb carrying the mRNA payload slips safely through the doorway and into the cell.
That is just incredible.
It really represents a paradigm shift in how formulation scientists must approach drug design. For years, the assumption was that to achieve higher stability and better cellular delivery, we had to invent entirely new, increasingly complex synthetic lipids from scratch.
Now we know that's not the case.
Right. What this research definitively proves is that the chemical environment dictates the internal architecture. Buffers are not passive packaging. They are active engineering tools.
And if we zoom out to the bigger picture, the real-world implications of this for everyone listening are profound. By precisely manipulating the buffer concentration leveraging that 150 millimolar trace environment, we can custom build highly stable lipid nanoparticles that survive the brutal physics of global shipping and ultra-cold freezing without losing their potency.
Which is huge for global health access.
Absolutely. This means life-saving therapeutics won't degrade just because a clinic's freezer fluctuated by a few degrees.
And this extends far beyond the prophylactic vaccines we are familiar with today. The future of medicine relies heavily on mRNA and lipid nanoparticle delivery. We're talking about advanced CRISPR gene editing therapies where precision is paramount.
Oh, wow. Yep.
We are looking at personalized oncology and cancer immunotherapies that teach the body to hunt specific tumors and protein replacement therapies for chronic genetic disorders.
All of those advanced interventions require incredibly stable, highly efficient delivery vehicles.
Exactly. And because these customized blebbed nanoparticles are vastly more efficient at escaping the endosome, clinicians could potentially administer significantly lower doses of the drug to achieve the same therapeutic outcome.
And lower doses inherently mean reduced cellular toxicity, less systemic inflammation, and a dramatically safer experience for the patient.
It's a win all around.
It really is. So to wrap this up, we started out wondering how the violence of freezing damages medicine and discovered that the liquid it flutes in can completely rebuild its internal anatomy.
Yeah, if there's two key takeaways here, it's really this. Number one, environment shapes structure, and structure dictates function.
Always comes back to that in biology.
Always. And number two, high concentration truss buffer acts as a biological architect, building blebs that protect mRNA and massively boost its cellular delivery.
But, this realization opens up a completely new, somewhat radical frontier of thought. If we can use simple environmental chemistry to force a microscopic fat bubble to fundamentally reorganize its entire internal anatomy and build itself a protective backpack, it makes you wonder what other hidden superpowers simple, everyday chemicals might have on the advanced nanotechnology of tomorrow.
It really is a fascinating direction to take this. Viruses have evolved for billions of years to use the subtle chemical shifts in our bodily environments to change their own shapes and trigger infections.
Right.
If we have proven that we can mimic that kind of structural shape-shifting using standard chemical buffers, we could theoretically move toward a highly personalized model of medicine.
But what would that even look like?
Well, imagine a future where buffers aren't just universally standardized at a manufacturing plant. What if, based on a rapid biopsy of a patient's specific tumor cells, doctors could custom mix a buffer right at the patient's bedside?
Oh, wow.
A buffer specifically calibrated to morph the lipid nanoparticle into the exact, precise geometry required to fuse with that individual's unique cellular membranes. We are no longer just delivering drugs. We are dynamically engineering the delivery vehicle in real time.
That is wild to think about. A bedside, custom-built nanobot, essentially.
Exactly.
Well, that is all the time we have for today. Thank you for joining us in the Deep Dive Lab. If you loved uncovering this scientific mystery with us, make sure you follow and subscribe for more fascinating science stories. Until next time, keep questioning what's beneath the surface.